Good afternoon, everyone, and it's my pleasure to welcome you to the 25th AIIMS Silver Jubilee Oration. Today, from the Ramalinga Swami boardroom at the All India Institute of Medical Sciences. I realize we're also going live on YouTube, and therefore we will have an audience that's both in person and online live. This oration was instituted when AIIMS completed 25 years of existence. We've had 24 previous orations prior to the one today. And this oration is typically awarded to an eminent scientist from either India or abroad, and not necessarily somebody who is a physician. It could be awarded even to somebody who's from any related specialty and doesn't necessarily have to be either a physician or even a biomedical scientist. The nominations are invited by the Institute every year, and almost anybody is free to nominate an individual. Their contributions to science, typically over the last 10 years, and particularly to research, what are evaluated by a central committee, which looks at all the nominees and then picks one for the oration. The oration itself carries a cash award as well as a plaque and is usually presented physically in person. But unfortunately, over the last few years, both with the combination of the COVID pandemic as well as our ability now to do it virtually, even though there is no pandemic, means that we are now able to do this oration virtually. And therefore, a number of orators possibly who may not have volunteered to be nominated earlier may be willing to be nominated because they may not need to travel to deliver the oration. Some of the previous orators, I will not narrate out all 25 of the previous orations, but some of the previous orators who may be well known to most people in the audience would be Dr. A.S. Paintal, Professor Salim Yusuf, Dr. M.S. Valyathan, Dr. Vinay Kumar, Dr. Jatin Shah, and of course, there have been 24 previous total orations. It's my pleasure also now to welcome the Dean Academic, Professor Subrat Sinha, and the Dean Research, Professor Rama Chaudhary, who will be chairing the oration today. The director, unfortunately, is not available. Normally, the director would also be available to chair the session. At this point of time, usually we would also welcome the orator with a bouquet, but since we don't have the orator in person, we will do a virtual welcome to the orator for today. That's Dr. Chandan Sen. Dr. Chandan Sen is the J. Stanley Battersby Chair and Professor of Surgery, the Director of the Indiana Center for Regenerative Medicine and Engineering, and Executive Director of the Comprehensive Wound Care Center at the Indiana University Methodist Hospital. At the Indiana University, he also serves as the Associate Vice President of Research and as the Associate Dean for Research. He's also the Executive Director of Comprehensive Wound Center and is recognized as a Lilly Insight Scholar. He completed his master's in physiology at the University of Calcutta and then obtained his PhD in physiology at the University of Eastern Finland. He did a postdoctoral fellowship at the University of California at Berkeley's Molecular and Cell Biology Department and has subsequently worked at the Lawrence Berkeley National Laboratory before moving on to Ohio State University and is currently at the Indiana University. His principal primary areas of research interest include tissue injury, repair, regeneration, and infection. His group has pioneered the tissue nanotransfaction technology in regenerative medicine, and the group received the 2018 Edison Award for Innovation. His work also led to the electroceutical management of tissue infection, and in 2016, this technology received the Frost & Sullivan Award for New Product Innovation. Earlier this year, Dr. Sen was elected a Fellow of the National Academy of Inventors. He has over 300 peer-reviewed publications, and it's my pleasure and privilege to invite Dr. Sen to deliver the 25th AIIMS Silver Jubilee Oration. Over to you, Dr. Sen.
Thank you, and a very good afternoon. It's early morning here, and it's quite a privilege to be a part of this distinguished group at this distinguished institution. And thanks to the organizers for placing their confidence in our team and our work. With that, in my association with AIIMS has been since 1989. There was the centennial meeting of the IUPS, International Union of Physiological Sciences. I was a student from Calcutta selected with a travel award from India. There was only one national award to students. And it is in Helsinki that I met for the first time Professor Manchanda, who was at the time the chair of physiology, Professor Usha Nair, who later on became the chair of physiology. And ever since, I've had a wonderful relationship with AIIMS, where I've learned much. And today I have wonderful colleagues at AIIMS in Professor Sujata Mohanty, who was in our lab like last month, Professor Amit Dinda, and many others. I'm also thankful to Dr. Pradeep Mahajan at STEMRX in Mumbai, who is working with us on the TNT project, about which I'm going to talk. And our current interaction with the group at AIIMS is funded by the Indo-US Science and Technology Forum. I express my deepest thanks to the IUSSTF. So with that, today, the nature of my talk will be more of a broad-based, trying to address the concepts and see how this field of regenerative medicine is unfolding. And because of the limited time, I'm going to focus on our technology, tissue nanotransfaction, that was just mentioned. And the goal here would be to cover a broad area to give a context as well as some specific details about the technology. Now, the field of regenerative medicine has been in the limelight for the last, I'd say, 2 decades. And there has been a lot of hype in the beginning, and as is with any new field, some disappointments, followed by some really interesting developments in science, following which some substantial concrete developments more recently that are likely to change the future of healthcare. As you will see from this slide, the US Food and Drug Administration has now instituted the RMAT track for or RMAT designation for product approval. At this time, if I recall correctly, it's about 67 or so products already approved. And this product is not just, so this designation includes cell-based therapies and several approaches where, to put it in lay terms, the future of medicine will not only depend on the so-called medicine that we know today and the devices that we know today, but simple body parts, more later complex body parts will be made and placed on hold, and will be plugged into the body as needed. And this is a huge transformation of the healthcare field, and I think this is a wonderful time to get involved in these developments. Now, as is true for any emergent field with a lot of potential, there's a lot of expectation placed, and then there's some failures. And the stem cell field is enormously, I think, powerful as a lesson in biology, in that when we went to school, we were taught about terminal differentiation, how certain tissues in the adult body, once differentiated, cannot seek or another fate or other functional fate. But now it seems that with the discovery of stem cell biology in the earlier days from fetal tissue, more later days, numerous adult stem cell niche have been found. It's very clear that we have these multipotent cells in the body, and unless we saw them as we see them today, we would not believe that these cells were actually present in the adult body. Now, the extension of that is actually even more interesting to us, which is stem cells obviously are quite prominent in displaying their stemness and their ability to acquire other types of functionality and fate. But then again, cells that are not stem cells, and I'll come to that shortly, also have substantial plasticity, and in some cases a more tempered plasticity, which makes these non-stem cell population more attractive to harvest with respect to therapeutic intent. I'm going to address that shortly. Stem cells have such high plasticity that indeed, for publication purposes, jaw-dropping data can be obtained, as is presented in numerous papers. But as we test the ability of these cells to address disease processes in the body, to keep a complete control of the entire population of the cells is quite challenging, meaning that some of the cells will become what we want them to become and will do what we want them to do, but there will be other cells that will escape our control and could end up being hazardous. And so that's a huge threat. And as we are learning more about stem cell biology, that threat will be successfully managed in due time. I think today is not that day. Now, as would be expected, the regulatory agencies are quite enthusiastic about this new development, but of course, they are keeping a very clear eye on safety before we talk about efficacy. So at this time, as we develop new therapeutic modalities, the focus on safety, especially if we have to test them in humans, is of paramount importance. Now, a quick snapshot at what we were taught when we were in school. We always knew that the embryonic tissue, was highly plastic. After birth, that plasticity seems to be dampened in a remarkable manner. And in adults, there is hardly much plasticity. Okay, and therefore we had the concept of terminal differentiation. What we know today is that indeed the embryonic tissue, fetal tissue is highly plastic. You go in utero and you cause injury, and you have types of regenerative repair that you will never see in an adult body. And this is quite well established. Now, if you go to newborn, it seems like that regenerative repair is substantially blunted, but you do see good repair happening. In the adult body, depending on the tissue in question, some tissues have low inherent plasticity. Other tissues have mediocre, inherent plasticity, and yet other tissues shown to the right with the pointer in the red have very high inherent plasticity. And among the tissues that have very high inherent plasticity are skin and fat. Okay, and we like skin because it is very accessible. It's a low-risk organ. If you made a mistake, you're not going to kill anybody. So we started. Therefore, we started with testing the inherent plasticity of the skin as a tissue, not just as a cell, but the tissue and an organ. Now, in general, why are we so excited about stem cells? Because stem cells will give us more stem cells. More importantly, stem cells will give us pretty much any specialized cells we want. Now, when we publish these reports, we end up showing exactly what we were hypothesizing and hoping to see. But what is true is that as we get what we need, we also have cells that are beyond our control, and in some cases could give rise to cancer cells. And this is absolutely, if you can benefit somebody, that's okay, but you cannot harm somebody. So unless we find a good way of harnessing the power of stem cells in a way that such hazardous outcomes can be confidently managed, I think this will stay an open area of investigation, and the benefits of stem cell as a whole will be viewed more skeptically until a more mature time of managing risk comes. Now, I think the developments related to induced pluripotent stem cells are fascinating. In this case, cells were taken from the skin, and then essentially they're for a few weeks in the laboratory, and they go through, as you see here, a number of steps. And once for example, if you started with the skin fibroblasts, in a few weeks, you now have all these pluripotent stem cells. And then the pluripotent stem cells can indeed be taken to almost any fate, given the body of the literature that we have today, except that all cells will not listen to us. There will be cells in the mix that will remain or retain their stemness. And upon injection into the body, of course, the cells that already differentiated to the cells that we were expecting to are likely to provide some benefits, but the cells that are not, submitting to this harness may escape and provide, some type of a cancer outcome, which is quite dreaded, and much work is currently going on to see how we can eliminate these cells that actually do not listen to us during this differentiation process. Now, to look at cell plasticity as a whole, just a very general schematic here, what you see is that you have embryonic stem cells. From these embryonic stem cells, you could get neural stem cells, you could get mesenchymal stromal cells, and those differentiated. But a very interesting finding that was reported a few years ago is that skin fibroblasts could actually be directly converted, in this case, as the shows, neuron without going through blindness. That's so important. So important because if you can that you need, for example, a new faint, achieve the without stemness, you do not with the of stemness of listen to us. Are very in, but we really thought single cell their own hardly do in a function. So our for reprogramming organ is over to sure, but this direct goal to develop technology tissue multicellular within the body, which is under immune illness, because if you take this out of the body as we do during IPS and put it in the lab, they become immunogenic. You put them back into the body, although it came from this body, the immune checks, and that's a problem. So in this case, if we were to be able to achieve tissue reprogramming in the live body, it would be happening under immune surveillance. And that's really, if we can achieve that, is a more sustainable solution than a quick solution that is publishable, but as it relates to functionality, the body constantly keeps fighting, and immune suppression, etc., are needed. Now, in this case, the reprogramming happens in negotiation with a complex in vivo microenvironment. In the laboratory, it's very easy to keep the incubator in a given setting, but you see in the body, there are diurnal variations. There are all types of complexities in changes that are happening over time, and those are not accounted for when we do reprogramming in the incubator. So therefore, that's another benefit of trying to achieve reprogramming in the body. And given the two above, this type of recipe should provide a more sustainable functional impact. So in this case, the tissue nanotransfaction technology, about which I'm going to talk, utilizes the body's own cells. So we do not isolate the cells, mind you. As you see here, this is a primitive form of the chip that is placed on the skin. And the nanochip delivers genetic cargo reprogramming the tissue as a whole. I'll come to that shortly. And we have published different phases of skin cells following this type of TNT, and this is uniquely novel. Now, the postdoc, Daniel Gallego Perez, who is the author of the first paper that we published in Nature Nanotechnology, won a Director's Award, NIH Director's Award. Daniel is now a highly competitive and a strong faculty at Ohio State in the Department of Biomedical Engineering. But after his award, Dr. Francis Collins at the NIH did this blog on TNT with the title "Skin Cells Can Be Reprogrammed in Vivo." And if you just go to YouTube and write tissue nanotransfaction, there are over 1,000 videos that have been made by different groups. But I'm going to share with you this video just to give an introduction to the concept of TNT.
As cells differentiate, they acquire specialized functions. For decades, scientists believed this process ended with terminally differentiated cells. Committed to a particular cell fate, the ability to switch function irreversibly lost. In a major breakthrough, iPSC technology established the possibility of inducing plasticity to terminally differentiated cells. But several limitations arose, including a lengthy multi-step lab-based process, possible inefficiencies due to experimental conditions, low fidelity with which the final cell type is generated, and safety concerns, including potential cancer risk. More recently, scientists found that mature, fully differentiated somatic cells could be directly converted to another cell type by ectopic expression of specific transcription factors. without the need to acquire intermediate pluripotency. However, this technique requires viral gene transfer, an approach which, 1, requires an exhaustive laboratory procedure, and 2, carries risks, such as insertional mutagenesis, irreversibility caused by stable transduction, and increased cancer risk caused by long-term proto-oncogene expression. Therefore, it is not well suited for point-of-care applications. Thus, a non-viral tissue nanotransfection approach was developed for direct cell conversion in vivo by Professor Chandan K Sen in collaboration with Professor James Lee. This work has been published in Nature Nanotechnology in 2017. This approach is simple, does not require elaborate laboratory procedures, and can be used as a point-of-care approach. This approach consists of 2 components. First, a hardware component, or chip, fabricated for efficient delivery of cell-specific cargo in murine model. Manufacturing of this chip has been scaled up to fit dimensions relevant to human need. Such chip has been successfully tested in preclinical porcine studies. Second, the cargo itself, which consists of a combination of different reprogramming factors. Thus, by applying a highly intense and focused electric field through arrayed nanochanels, this device then operates the adjacent tissue cell membranes and electrophoretically drives reprogramming factors into the cells. This new nano-electroporation technology is distinctly different from bulk electroporation. The simplicity and utility of this approach was demonstrated by nano-electroporating the skin that served as an agricultural land to cultivate cells of different functional abilities. Using a specific cargo, a combination of different reprogramming factors, skin fibroblasts were successfully converted to endothelial cells that ultimately formed blood vessels with 7 days. These blood vessel networks anastomosed with the parent circulatory system and restored tissue and lymph diffusion in 2 murine models of injury-induced ischemia. Similarly, using a different cargo, skin fibroblasts were converted to neuronal cells that showed similar gene expression profile and electrophysiological function to that of cells in the developing brain. This tissue nanotransfection platform technology has the potential to ultimately enable the use of patients' own tissue as a prolific immunosurveilled bioreactor to produce autologous cells that can resolve conditions locally or distally upon harvesting. This simple-to-implement nano-electroporation technology generates favorable biological responses. It is a one-time treatment, applied topically, and only lasts for a fraction of a second, without elaborate laboratory procedures. And it could have applications beyond reprogramming-based cell therapies, such as gene, protein, and drug delivery.
Thank you for your attention to that video, and I hope the concept was clear enough. Now, this is the original paper that was reported 5 years ago. All the data I'm going to show today, about 20% was reported 5 years ago. About 60% was reported in the last 2 years, and 20% is not reported. That's the distribution of the data that I'm going to show you. After this was published, there was an editorial in JAMA, Nanochip turns skin into a bioreactor. And there was also an editorial in Science Translational Medicine, Nanotransfaction brings progress that's more than skin deep. And there's been a lot of interest in this technology as it relates to the simplicity and the types of outcomes it's able to achieve. So what exactly is this technology? Very briefly, so we know about bulk electroporation. Now, when you do bulk electroporation, what happens is a vast number of cells die. Some cells don't die. Cells that don't die have a hole in their membrane. And then when you put some plasmid outside the cell, they get inside the cell, these membranes are repaired, and now we have gene delivery. But what also happened in the meanwhile is that these holes caused in the membrane causes membrane scarring and dampens the inherent plasticity of the cell. So we were interested in finding a very pointed approach to deliver the goods, but not cause this type of cytoskeletal scarring as we currently are studying. And in the example that you see below shows that 2% of the cell surface area is open to deliver using the nanotransfection approach, and you will see how, in this way, a very directed or deterministic delivery of cargo can be achieved. I'll spare the technical details, but you get the idea that compared to bulk electroporation, this is a much more sophisticated approach where the membrane injury is minimized, and therefore the inherent plasticity of the cell is retained. Now, we recently reported there was a lot of interest in the chip. A lot of groups were asking, and I was not able to provide to most groups that were asking. So we decided to publish a 53-page article that details the production of the chip. The article just came out in Nature Protocols last year, and Professor Yi Zhuan, who is currently leading our nano technology group, spearheaded this. He's currently a K25 awardee of the NIH under my mentorship. And as we actually have this type of electric field pass through a tissue matrix, there are very important complex modeling considerations here that are critical with respect to understanding where these types of vectors that we are delivering would rest. So depending on whether if you want the delivery to be superficial, say in the epidermis versus dermis versus sub-Q, you can actually tweak the physical parameters and get these tissue vectors in the right place only if you knew how their path would be through the tissue. And for that, we published this modeling paper that guides and provides the equations to guide our therapeutic purposes. These are all, Lee published this in 2022 from our group. Now I'm going to show you a little bit about the chip.
Certain organs in our body can serve as a bioreactor. Through real-time feedback from our immune system, we can grow new body parts. We can even grow them in one place and then transplant them to where they are needed. One well-suited organ for such bioreactor function is the skin. This largest organ of the body provides ample real estate to grow therapeutic functional tissue structures. Tissue nanotransfection technology, or TNT, enables in vivo reprogramming of the skin to produce functioning blood vessels, active nerve tissue, insulogenic tissue, and more. This process depends on specialized chips created from silicon wafers. A coating is added to the top, then lasers are used to write small holes into the chip, then etched deep into the wafer. On the other side, a new coating is added and then lithographed to create hollow microneedles. The chip is then etched to make the needles longer, clean, and then treated with a new coating to improve precision. The exact nanofabrication process for manufacturing the TNT chip is now reported in a new publication in Nature Protocol. The delivery mechanism consists of two components. First, the chip, fabricated for efficient delivery of cell-specific cargo. Second, the cargo itself, which consists of a combination of different reprogramming factors. The cargo is different for different applications. By applying a highly intense and focused electric field through arrayed nanochannels, this device then operates the abutting tissue cell membranes and electrophoretically drives reprogramming factors into the tissue. In this approach, the skin may be viewed as an agricultural land that can be used to cultivate cells and multicellular structures of different functional abilities, such as perfused blood vessels, electrophysiologically active nerve tissue, etc. Using a specific cargo, a combination of different reprogramming factors, skin fibroblasts were successfully converted to vasculogenic cells that ultimately form perfused blood vessels within 7 days. These blood vessel networks anastomosed with the parent circulatory system and rescued against ischemic insults. Similarly, using a different cargo, neurogenic as well as neurotropic reprogramming skin cells were achieved. Such intervention proves to be promising to rescue against stroke as well as diabetic neuropathy. In future, TNT is expected to involve dispensable chips with injectable factors supplied as liquid-loaded cartridges. During use, the chips will be stamped on the skin. Such approach will be compatible with point of care using minimum training for local and remote use. This TNT platform technology has the potential to ultimately enable the use of the patient's own tissue as a prolific immunosurveilled bioreactor to produce tissue, grow body parts, and resolve conditions. and is a one-time treatment applied topically, only lasts for a fraction of a second, and can be applied at point of care without elaborate laboratory procedures. And it could have applications beyond reprogramming-based cell therapies, such as gene therapy, protein delivery, drug delivery, and cell-specific exosome labeling.
I'm going to now share with you some of those applications just to give you a real-life idea of what has been achieved so far and what is in process. First, I'm going to talk about the TNT that is vasculogenic. So the first example you have to your right of the screen. And this was achieved by Durba Pal. Durba is now an associate professor at IIT Ropar. Shivadip, all three were my postdocs. Shivadip is now an assistant professor. And Kanaya Singh from BHU is currently an assistant professor in the US. And Dr. Khatak also from the PG Institute in Calcutta is an assistant professor here. So in one example, so this paper is already out, so I'm just going to give you some concrete examples. So you have the femoral artery, 1 centimeter of the femoral artery has been transected, and then ligation has happened. And after 3 days of this surgical procedure, TNT EFF, are three plasmids, E2V2, FLE1, and FOXC2, that were then transferred into the skin. And in about 14 days after that, you start seeing, if you look at the TNT mark to your left, you will see trickling blood flow in the superficial microcirculation of the skin that is expected. In the TNT, in 2 weeks, you start seeing pulsatile blood flow in the superficial 3 millimeter of the skin. You then do histology, and then you trace using transgenic mice to show that the skin cells are now participating in functional vasculature and rescuing the limb. So you see to your right with laser spectral imaging, the blood flow is substantially increased. So you see to the left the TNT mark. There is auto-amputation. The limb pretty much falls off because of such severe necrosis. You have a pink leg with the animal running around. We have videos of the animal running around without a femoral just because of a 0.1 second procedure done only once. Okay, this procedure cannot be done twice, at least for all these experiments, only done once, 0.1 second it lasts. We then took this tissue where TNT was performed, and we found evidence that what we were doing, the active principle, was getting actually encapsulated in exosomes and was getting dispatched by the skin. So we took the skin out and we collected the exosomes and we injected the exosomes into the hind limb, the same type of hind limb surgery done, and then in this case the EVs were injected 3 days after hind limb surgery. And we were able to get complete recovery of these limbs. So you can do it TNT directly, or you can just take the exosomes from this tissue, post-TNT tissue, and inject them into the limb. That will also provide the types of benefits. And these are already reported. More recently, Daniel's group, where I have also participated, showed that in a sciatic nerve crush model, that TNT EFF was very effective in rescuing the crushed nerves. Now, a few years ago, a decade ago, when Cyrus Chang was in my lab, he's currently back in Hong Kong, we actually stumbled upon a very interesting finding where we found that when you injure the skin, there is sharp of the healthy skin, not the diabetic skin, the healthy skin, there is sharp downregulation of MIR200B. And then we found, as was reported in this paper 10 years ago, at that time, our concepts of tissue reprogramming were not as mature as today's. So we thought that MIR200B, as we reported in this paper, was an angiostatic agent that had to be taken down for the wound angiogenesis to happen. Little did we know, and as we know today, that the injury itself is actually a reprogramming stimulus. So we now see that when a tissue is injured, automatically physiological processes are unleashed by the downregulation of MIR200B, which then results in the conversion of skin fibroblasts to what is known now as, we call them vasculogenic fibroblasts. There is a paper on identification of vasculogenic fibroblasts coming out. We expect to be out in a major Nature journal within a couple of months. So mid-200B, we found out, was not an angiostatic agent. It actually had to go down to unleash a tissue reprogramming process, which then would convert skin fibroblasts into vasculogenic fibroblasts, which then would actually lead to the formation of new blood vessels. So, of course, we were interested. So that happens in the healthy skin, but does not happen in the diabetic skin. Okay, it does not happen in the diabetic skin. So the diabetic skin, we had to force it. And the way we forced it is by actually generating these antisense oligos. So you will see in the future slides ASO, and that ASO stands for antisense oligos. And in this case, the antisense oligo is designed as a mid-200B inhibitor. So what you see here is that in gene therapy, viral or vectors are of concern. And in general, from a regulatory standpoint, using antisense oligos seems to be far more desirable than using some other forms of gene vectors. So if you look at the single cell sequencing data of ASO-treated human dermal fibroblasts, what one would see, if you look at the panel to the bottom, you will see that cluster 1, shown in green, is very little in day 0, but in day 1 and over 7 days, you see a lot of cluster 1 cells. Okay, and this is where we've identified the vasculogenic fibroblasts. So you have all these clusters of fibroblasts, and there are all these other clusters that do not move. But there's a particular subcluster of fibroblasts that actually responds to the injury and acquires vasculogenic characteristics. So if one takes a quick look at the example, if you look at the panel above, we show cluster 0, 1, 2, and 3. All those clusters are also shown to the top right of your screen. And if you now look at only cluster 1, which is of particular interest to us, you will see that in this cluster, there is a loss of collagen-related genes, as would be more fibroblast-related over time, and acquisition of endothelial-specific genes like VEGF-B, VEGF-C, neuropilin, so on and so forth, starts getting acquired over time. Now, if you now take a look at this very specific cluster over a period of time, you will see COL1A2 is a signature gene characteristic of the fibroblast, and you will see a loss of COL1A2 as shown in blue in the top panel, and a gain of VEGF-B shown in blue in the bottom panel in that cluster 1 that we have shown in the previous slide. So with this, what we have then found is when we did this trajectory analysis through pseudotime analysis, we started finding that there's a novel fibroblast subpopulation. So there are two important operative words here. First of all, not all fibroblasts are open to such reprogramming. It's a subpopulation, point number 1. Point number 2, it is not what other people call, as some people call it, as induced endothelial cells. Induced endothelial cells would mean that all fibroblast characteristics are shed and endothelial characteristics are acquired. But what we see here is not induced endothelial cell. What we see here are cells that retain certain fibroblast characteristics and acquire new vasculogenic characteristics. So we refer to that as a state change as opposed to a fate change. Okay, in a fate change, the cell would have to lose its characteristics and acquire the new characteristics. So here, what you see is a state change acquired by a subpopulation of skin fibroblasts. So that's on the vasculogenic side. A quick example on the neurogenic side. So Dr. Khanna's group leads this project now. So if you look at day 1 and you do TNT with ABM. So in this case, ABM is the neurogenic cocktail, as shown to your top left, ASCL1, BRN2, and middle 1. So day 2, you see day 2 and 3, you see delivery of the plasmids. In day 5, you start seeing TUJ1, MAP2, and TUJ1 is like a neuron-specific class 3 beta tubulin. MAP2 is macrotubule-associated protein 2. These start showing up in day 5. And then in the third week, you start seeing electrophysiology. of those neural specific electrophysiology of these cells that you reprogrammed. If you waited until 8 weeks, so please note with arrow the timeline, in 24 hours, you see the delivery of the plasmid. In 4 weeks here in vivo, you see a lot of TUJ1 positive cell in the skin, not supposed to be there. Now they are there. And you see there are not one or two. There are tons of TUJ1 positive cells in the skin. And then you see in 8 weeks, you start seeing neurofilament, you start seeing synapsin, you start seeing choline acetyltransferase, which you know is the enzyme responsible for the synthesis of the neurotransmitter acetylcholine. So if you do a patch camp-like study, as we have reported, you can actually show that these skin cells are now excitable and they share the electrophysiological properties of the neuron. And you have thousands and thousands of them in the skin. You can actually harvest that piece of skin and get all these tissues out, the neural cells out, and you can plant them back into the stroke-affected brain, and you can recover brain function. We have published that. Now, interestingly, while you have this neurogenic reprogramming happening, as you know, you saw TUJ1 cells, but then you saw some cells that don't show TUJ1, right? So what I'm trying to show you here is the cells that don't show TUJ1 positive are also affected in that we found they start making large amounts of neurotrophic substances. So what we did is, when you now go to the diabetic limb and you do this type of a neurogenic TNT, you get a neurotrophic environment that is very helpful to rescue against diabetic polyneuropathy. Because here you have the nerve fibers in circuit, and it is about rescuing those nerve fibers by actually harnessing the power of the neurotrophic environment in the skin caused by the neurogenic programming of TNT. Dr. Manishekar Kumar is now working on something that we have not yet reported. This is a very high interest, commercially important project. So this is again the chip with an insulinogenic cocktail touching the skin for only 10 seconds once. And once you do that once, you start seeing copious amounts of C-peptide and insulin to a point where over a period of the next 9 weeks, only once, 1/10 of a second, the chip touches the skin. For the next 9 weeks, you have benefit in blood sugar and blood glucose. You also have IBGDT, and because of the extraordinary results, we got an NIH grant, and we just started working with Wake Forest, and we did 2 independent studies on the monkeys. And these are diabetic monkeys, not experimentally diabetic, naturally diabetic monkeys, and the results are very encouraging. So right now I cannot get into much details about it, but just to say that insulinogenic reprogramming is something that will come out from our lab pretty soon. The next example is a paper that got accepted last week, is the model of volumetric muscle loss. And in this model of volumetric muscle loss, now we actually went and did not TNT of the skin, but TNT of the skeletal muscle. And by doing TNT of the skeletal muscle, we were able to achieve myogenesis, and that, so following VML, improved torque development and also improved muscle weight. This paper just got accepted in Nature Regenerative Medicine and should be in PubMed maybe in a week or two. Finally, this paper that just came out, Kanaya Singh joined us some 3 years ago from BHU, and this paper just came out in JCI, and he was able to use TNT in vivo to achieve cell-specific gene editing. So he studied a whole lot of chronic wound patients to find that in this chronic wound patient, because of the biochemical microenvironment of the wound, you have massive hypermethylation of P53, which is a major problem. So he went ahead and developed a method, paper is out, so you can read that, where you can actually achieve CRISPR-Cas9-based P53 in the keratinocyte of this wound edge, and was able to close these wounds. So that's a work that is already out. In the next example, this is Professor Gail Gordillo, who visited AIIMS a few years ago in 2016, and Dr. Singhal, accompanied her, and she's our chief of plastic surgery, and she's interested in hemangioma. And in this hemangioma story, so she collaborates with Professor Roy, who is an expert in macrophage biology. So the two of them collaborated to use TNT, and this paper should be coming out also very shortly. In short, it was seen that tumor cells actually dispatch tumorigenic factors in exosomes that goes and makes the macrophage tumorigenic. The macrophage becomes tumorigenic. And this process can be interrupted by TNT and tumor regression can be achieved. So you see here that all the animals are dead within, say, 15 or 16 days, as shown by the Kaplan-Meier curve in blue. And in the case of the TNT MIR126 inhibitor group in red, you see how these animals keep going, and you see the tumor regression at, that is shown on the panel to the right, and at the panel to the bottom, the blockade of this MIR126 pathway, which is the causative angiomere that causes this type of tumor, is also shown here. So this paper should be out in Molecular Therapy also within the next 1 month. While this paper was going on, what you see here, is a nano imaging of the exosome. So you see localization of MIR126 in the exosome, and CD81 here is marking that these are exosomes. And then there was, in this particular approach, there was a targeted approach to take down these MIR126 in the exosomes, and complete tumor regression, as shown, was achieved. Finally, this paper came out, I think it was last year or the year before. Dr. Ghatak's group has come up with a way of using TNT to color label, fluorescently label cell-specific exosomes. So you actually have the keratinocyte originating exosome in green, the endothelial originating exosome in red, the fibroblast originating exosome in some other color, and you could actually see the dynamics of exosome trafficking in the live body. So should you be interested, this paper is out, you may take a look. And using this approach, he was able to show that in the wound healing process, there is a crosstalk happening between the fibroblast and the keratinocytes of the wound and the macrophages of the wound. And if you were to intercept that in the wrong way, the wound healing would be impaired. And if it is already impaired in diabetes, as he showed, you can correct it, wound healing would resume. So this means that exosomal assisted crosstalk between host cells and visiting cells like the wound macrophages are critically important. In the last example, I think I have, or this is the second last example. This is the case of lymphedema. So you have here mastectomy. One of my mentees, Al Hasnain, is a plastic surgeon that does this mastectomy, and he sees that 1/3 of his patients have lymphedema of the hand. And what happens is, if you look at the panel B, these vein and the lymphatic trunk are juxtaposed. And in developmental biology, there are 2 genes that are responsible for the cleavage of the vein and the lymphatic as separate chambers. And if you can undo that cleavage, you can actually achieve lymphovenous anastomosis. Today, lymphovenous anastomosis in patients are achieved surgically. It's a very cumbersome process, effort-requiring process. In this case, Al was able to develop a technique based on TNT that would actually cause lymphovenous anastomosis by the tip, by the chip touching the tail. Of course, it has a particular set of plasmid that he delivers, and in this doing this, he was able to achieve relief from lymphedema. So this is an experimental study that a part of which have been published, and Al is now funded with a few grants and is working on this. So this is lymphedema solution by TNT. The last example I'm going to show you just was published last week, where in this case, topical TNT was actually used to uncover, we have this, like I talked at the outset, perfect fetal repair, regenerative repair, when we are in the fetal part of our lifespan. When we come to the adult part, we seem to have lost it. And of course, when we have diabetes, then even the adult normal physiological wound healing is impaired. So in this case, Jubilee was able to show that using TNT, you can actually re-engage a fetal pathway in the diabetic adult. So re-engage a fetal pathway in the diabetic adult. How was this achieved? This is already out. And the origin starts with a factor coming out of the blood clot. And this paper would be of interest. In the interest of time, I'm not going to get into the details. Finally, I'd like to communicate greetings from our institute and from our school, Indiana Center of Medicine and Engineering at the School of Medicine at the Indiana University. And I'd like to thank all the sponsors, primarily the NIH and the U.S. Department of Defense. Thank you very much for your time. Of course, this time when I went to India, I was in India last week, and many of you told me that going to US is a problem. So I came back and I asked a few people around. The visa delay that you see while coming to the US is primarily for tourist visa. The J1 visas are still on time. So if there's anybody who's interested in learning more about TNT and perhaps doing a postdoc, we have many available. Do let us know. Thank you very much for your time.
Thank you. We would now like to make a presentation of the plaque and the certificate. Typically, again, it's done physically where the director would present it to you, but we do it virtually as well. We've learned how to do this. So we'll present it to you and show it to you what it looks like. So maybe the dean would show this is the institute plate, which we would like to present you. We'll ensure that it reaches you somehow, maybe on your next visit to the institute.
Thank you very much. Thank you, sir. Really appreciate that.
Can you show the camera there?
Are you able to see me? Yes.
Yes.
So,
thank you. Wonderful. Wonderful. Wow.
It's wonderful.
Very impressive. Thank you.
And we've just missed you by Dr. Rama. Come here, please.
So we
You are
actually
Dean
missed only
Research,
11th, I
and
think.
yes, and I'm the Dean Academics. And we've just missed you by a few days. I know you were here last week, but anyway, I think this way we'll have the pleasure of your company in our institutions once more. So that's the
Absolutely.
good part of it.
Very much look
Thank
forward
you very
to my
much
honor. Thank you.
for agreeing to accept this oration.
Yeah.
Thank you.
And this is this is a certificate that we have in case, so
Thank you. Thank you.
thank you.
So as we come to a close of this oration, I'd like to thank Dr. Sen for agreeing to deliver this oration, for accepting an invitation. And like Professor Sinha said, maybe we'll have another talk when you're here the next time to make up for the physical absence this time. I'd also like to thank the Institute, the Deans, and the All India Institute of Medical Sciences for the institution of this oration and for giving us the opportunity to have this today. Members of the research team, the Dean Research and the Associate Dean Research, who made this a part of the research day. This is actually the beginning of our research day. The research day itself is tomorrow, but this is a part of the research day celebrations as well. So thank you for making this a part of the research day celebrations. To all the faculty, staff, students, researchers, everybody who's here physically, as well as everybody who's watching this online, for taking time out and for being a part of this oration today. Thank you, everyone.
Thank you again. And as I close, I'd like to also extend my thanks to Dr. Manish Sengel and to the numerous colleagues and students that I work with at AIIMS. You have taught me much, and I very much look forward to go back and learning more. Thank you.
Thank you. And for everybody here, we have tea being served in the lounge outside. You're all welcome.